Hello! In this lecture, we'll be continuing our discussion of the flagellated protozoa that infect humans, and we'll also cover the major amoeba that cause human parasitic disease. The first parasite that we'll discuss is Giardia duodenalis. This parasite actually is referred to by at least 3 different scientific names, and there isn't widespread agreement about which species name is correct. So you'll see references to Giardia duodenalis, Giardia intestinalis, and Giardia lamblia. You'll see that in the textbook, you'll see it in the literature. So for our purposes, we'll just make note that all 3 designations are acceptable. So Giardia is the causative agent of a disease that's referred to as giardiasis. A more colloquial term for the disease is beaver fever because beavers are an important reservoir for the infection. The life cycle is relatively simple and consists of just 2 stages, 2 major stages. On the left, you can see the active replicating form that has a high metabolic rate, and that form is called the trophozoite. This trophozoite is very characteristic. Once you develop sort of a search image for Giardia, you don't confuse it with anything else. These structures here that look like a couple of eyes are actually nuclei. So you see a nucleus with a nucleolus, another nucleus with a nucleolus. And it's a little difficult to see individual flagella, but there are several pairs of flagella that are coming up in the anterior end, and there's a couple of flagella that extend down the posterior part of this cell. So keep in mind, this is a large but single-celled protozoa. This darkly staining body here is known as the median body, and it really doesn't have any particularly known function. On the right-hand side of the figure, you see a cyst stage. So in the intestinal tract, as these organisms move down the digestive tract and undergo dehydration, a cyst stage is formed. And here is an early stage cyst where you still see the 2 nuclei that were present in the trophozoite, and you see remnants of the flagella extending across the center of the cyst. This is a very characteristic oval cyst, and this is the infective stage of Giardia. This photograph shows you a scanning electron micrograph of Giardia, and what you can see is that there is a large adhesive disc, which is not necessarily visible in the previous photograph. So each of these is a trophozoite stage, and you can see the flagella that are associated with this stage. And you can see that these parasites use this disc to attach firmly to the mucosal surface in the small intestine. So in general, the trophozoite is rounded at its interior end and pointed at its posterior end. It is somewhat dorso-ventrally flattened, being convex on the dorsal side, and on the ventral side you have that rather concave adhesive disc that allows for attachment to mucosal epithelial cells. As I stated, there are multiple pairs of anterior flagella, 4 pairs to be exact, so 8 total flagella that arise from kinetosomes in the anterior region of Giardia. There are 2 prominent nuclei, and the median bodies that are usually darkly stained in a fecal sample are prominent but of unknown function. As I said, the trophozoites attach to the intestinal cells using that disc. They feed on mucus secretions, and they reproduce asexually within the small intestine. So please note that these organisms are feeding on mucus. They are not invasive. They do not disrupt the mucosal membrane, and therefore the pathology that you see is predominantly due to the presence, the physical presence of these organisms, and the inhibition of absorptive properties that occurs because of their presence. In heavy infections with this organism, the entire mucosal surface of the small intestine can be covered by the parasites. Here is a scanning electron micrograph that shows a small region. You can see some of the underlying small intestine, the mucosal cells, and you can see literally in this image alone dozens of the trophozoites. The previous picture that I showed of a cyst had 2 nuclei, but as the cyst develops in the environment within the cyst wall, eventually it will develop 4 nuclei when it's completely mature. The median bodies and the flagella are very often visible in a stained specimen, and there's always a thick, usually hyaline. It looks sort of transparent. The previous picture that I showed you didn't necessarily show that attribute, but other figures that you'll see a little later will. It is the cyst that is the infective stage, and mammals acquire this stage when they swallow the cyst in food or water. And the cyst is swallowed, it passes through the stomach where it is acted upon by acids in the stomach as well as the low pH. And then the cyst wall is ruptured and the trophozoite stages escape and establish the infection. Now, keeping in mind that Giardia is replicating within the small intestine and sometimes up in the villi of the intestinal tract, these are in somewhat of an anaerobic environment, not completely oxygen-free, but certainly under low oxygen environments. And Giardia has adapted to this environment by being what we call an aerotolerant anaerobe. And what that refers to is the fact that they can survive in the presence of oxygen, but they do not use oxygen for cellular respiration. In addition, very interestingly, Giardia lack mitochondria. They actually lack a variety of typical eukaryotic organelles. And because they lack mitochondria, obviously they can't carry out the Krebs cycle or electron transport oxidative phosphorylation. And what this means is that their ATP production is limited to glycolysis followed by fermentation. And Giardia carries out alcoholic fermentation. During this process, if you think back to your basic biology of metabolism, you know that CO2 is liberated during alcoholic fermentation, and it's actually the release of CO2 gas that causes the bloating and the flatulence that's associated with giardiasis. So the clinical symptoms then do include watery, oily diarrhea, which is never bloody, so it is not dysentery. The amount of diarrhea can be pretty abundant depending on the severity of the infection. The patient typically experiences abdominal pain, bloating and flatulence associated with CO2 production by Giardia, nausea, and sometimes vomiting. And as I stated previously, the sheer physical presence of the abundant trophozoite stages in the small intestine interferes with the absorption of fats and other nutrients and causes malabsorption. And in chronic infections that persist for a period of weeks, you can see a pretty significant weight loss and pretty significant morbidity. Just to clarify the term morbidity, morbidity refers to disease and incapacitation. So we typically talk about morbidity, which is the illness, and mortality, which of course is death. And really, giardiasis is not associated with any significant mortality. The only danger of giardiasis would be through dehydration if someone was not treated properly. Transmission then occurs because the trophozoites detach from that mucosal epithelium. They move down the digestive tract. As the feces enter the colon, dehydration is occurring and the cyst stage forms. The cyst is released into the environment along with the feces, and the cyst is the infective stage. One of the things that is important to the epidemiology of giardiasis is that symptomatic patients are not releasing cyst. So the people who are the most ill are not the ones that are the most contagious. The reason for this is because someone that has a fulminating diarrhea is releasing trophozoite stages and not cyst, because there's no opportunity for dehydration and cyst formation to occur. They're releasing abundant trophozoites into the environment along with their stool, but trophozoites are not infective because they cannot survive passage through the stomach. So that means that it's the asymptomatic carriers, the people who are not symptomatic, that are responsible for transferring this infection. Similarly, reservoir host mammals in the environment are also asymptomatic, but they are releasing cyst into the water, which an unsuspecting person can come along and drink and become infected. Giardiasis is found across the world, and we certainly have it in North America. And if you go to the CDC website and you explore the disease, what you'll find is that the vast majority of cases have occurred when people drink surface water contaminated with the cyst stages. So a very common scenario is people are backpacking and they come across a beautiful clear stream and they decide to just dip in and take a drink. Children and adults have also acquired giardiasis by swimming in public swimming areas, and especially where there are lots of people sort of concentrated in a relatively small area. And if the cysts are there and you're splashing and doing somersaults and swallowing water accidentally, then you can become infected. It's actually fairly generalist in the mammalian host, and almost any aquatic mammal like beavers and muskrats and otters can become infected. Also raccoons and deer, and almost any mammal that drinks the cyst can act as a reservoir. In terms of the protective immune response to Giardia, this is an extracellular parasite that replicates in the small intestine and is attached to the mucosa. So consequently then, really only the humoral response can be a protective response. So we're talking about antibodies. And in addition, because this is strictly in the digestive tract, and these organisms are actually consuming mucus, IgA is the most important class of anti-Giardia antibody. We do know that when people are exposed to giardiasis, that anti-Giardia IgA is produced, and in very large measure, it's the production of these antibodies that allow the patient to resolve the infection over a week or two. Now, just to remind you, the humoral response is supported by the CD4 positive Th2 lymphocytes that are releasing cytokines such as interleukin-4 and interleukin-5. These cytokines support B lymphocyte activation, B lymphocyte replication, B lymphocyte differentiation, and antibody synthesis. And interleukin-5 in particular is known to cause the heavy class switch from mu to alpha, which is going to allow the plasma cell to produce IgA instead of IgM. We do know that Giardia does undergo some antigenic variation, and so there are different strains of Giardia that express different surface antigens. There are about 180 or so different antigenic types that have been identified. It's not the same type of antigenic variation like you see in the African trypanosomes. It's more similar to the antigenic variation that you see, for instance, in influenza. Over time, in endemic areas where people are frequently exposed to Giardia cyst, it seems quite clear that clinical immunity develops and people stop developing symptomatic responses when they ingest Giardia cyst. The disease is diagnosed by observing fecal samples and identifying either trophozoites or cyst in the fecal samples. Sometimes it is difficult to do this because the number of cysts or trophozoites might not be particularly high at the time that you are examining the stool. So usually you have to look at multiple stool samples, and they are trying to use more sensitive techniques to find rare cysts or trophs that might be present. And the indirect immunofluorescent assay is one assay that's used, and also PCR. The standard treatment for giardiasis is metronidazole, and metronidazole is sold by a couple of different trade names, and Flagellated is probably one of the most common. The other flagellated protozoa that I wanted to mention, and the last one, is Trichomonas vaginalis. And Trichomonas is in the family Trichomonadidae, and the disease is known, that's caused is known as trichomoniasis. And this sexually transmitted parasite has a cosmopolitan distribution. It requires intimate sexual contact. Since there is no cyst stage, there is only a trophozoite stage that has to be directly transmitted from one individual to another. So it's a very simple life cycle, and there is only one stage, and it is the trophozoite stage. To describe the trophozoite stage, there are 4 anterior flagella with a 5th flagellum that curves back along the cell body and forms an undulating membrane. Recall the undulating membranes that were present on certain stages of the kinetoplastid flagellated protozoa. Trichomonas vaginalis also has a prominent axostyle, which we learned about in the previous lecture. And this axostyle is really just a bundle of microtubules that runs along the length of the cell body. It's thought that that axostyle gives some support, rigidity, and maybe even a little bit of flexibility to the trophozoite. The trophozoite has just one nucleus, and it's usually very clearly obvious in stained samples. The Trichomonas trophozoites also have hydrogenosomes, and you may recall that the hydrogenosome is a special microbody in which you get ATP production and the release of hydrogen. So the hydrogenosome then allows for sort of extra ATP production using sort of unique chemical reactions. In addition to the hydrogenosome, which is really more of a supplemental ATP production, the principal way that Trichomonas vaginalis produces ATP is by glycolysis, followed by lactic acid fermentation. Here is a photograph showing the trophozoite stage, and you can see the major features that I mentioned. Here's a prominent nucleus. Here is the axostyle. Here are several flagella that are coming out of the anterior end. They're kind of bundled here in this image. Here's one coming off this way, and here's a couple. Here's the 5th one that is attaching to the cell body and forming this undulating membrane. The disease, known as trichomoniasis, occurs because of the replication of the trophozoites, typically in the vagina and urethra of women. Women are very much more likely to experience clinical symptoms than men. In men, trophozoites are located in the prostate gland, in the seminal vesicles, and in the urethra. And in terms of the epidemiology, the asymptomatic men are extremely important in keeping this infection going and passing it on to sexual partners. The strains that are pathogenic do tend to cause pretty intense inflammation and even some erosion of the vaginal epithelium in women. And because of that physical damage or erosion of the epithelium, you do get an intense leukocyte infiltration or inflammatory response. The inflammation and tissue damage actually makes women more susceptible to other STDs like chlamydia and gonorrhea and HIV. Some other symptoms include a profound itching sensation and a copious whitish discharge from the vagina, and that discharge, when it's examined under a microscope, is literally teeming with mobile trophozoite stages. One other feature that I don't have listed on the slide is that when the cervix is examined, there are often small punctate red lesions, and this is sometimes referred to as strawberry cervix. Once again, not 100% of the time, but most of the time, men do not show clinical symptoms and yet are very infectious. The infection, as I indicated, is diagnosed by microscopic examination of either urethral, vaginal discharge, and the obvious presence of these moving trophozoite stages. Metronidazole, once again, Flagyl, is most commonly used, and obviously it's really important to the epidemiology of this disease to identify and treat any sexual partners.